Hi everyone, my name is Alda Cahyaningtyas and this is my presentation on immune response. We're going to start with the entry of the antigen. The antigen typically make their entrance through the skin or mucous membrane. The skin and mucous membrane are part of the first line of defense. The mucous membrane are present in the respiratory, digestive, urinary, and reproductive system. Entry of antigen can happen when the pathogen have successfully penetrated the layers of the skin. However, since we do have that first line of defense, the body can essentially still fight this antigen. The epidermis is facilitated with dendritic cells. The dendritic cells are able to perform phagocytosis that is non-specific, meaning it is able to perform non-specific innate immunity. It is not mentioned here on the slides. However, the dermal layer or the dermis layer also provides structural role in the innate immunity. The dermis layer contains collagen, and collagen is tough fibers of protein, and essentially that gives the skin strength in order to withstand itself from pathogens trying to enter the body. Aside from providing structural innate immunity, the dermal cells also produce several chemicals that have a role in the chemical innate immunity. For example, the antimicrobial peptides such as dermcidins, they disrupt bacteria structurally, causing them to die. And then they also produce an enzyme called lysozyme of the sweat, which destroys bacterial cell wall, which can lead to osmotic shock and phagocytosis. The sebaceous glands of the skin also secrete sebum. The sebum actually lowers the pH that's on the surface of the skin, which prevents the growth of pathogens on the surface of the skin. And then the stem cells produce goblet cells to form barrier with the mucus that's secreted by the goblet cells. As the infection progresses, the second line of defense will start to come into play. The plasma, which is the liquid portion of blood, is a protein that will form blood clots to reduce the chance of blood loss and also reduce the risk of infection. It is a form of a defense mechanism. Transferrin and ferritin are both transport proteins. They have a role in transportation and storing iron for the functions of the human body. However, they also have a secondary role, which is to make iron unavailable from microbes. Leukocytes or white blood cells, such as neutrophils, eosinophils, and macrophages, are able to go through diapedesis. Diapedesis is the process of these leukocytes moving out of the blood vessels in order to perform phagocytosis to the antigen invading the body. Another leukocyte, which is basophil, they can also perform diapedesis, however, they do not perform phagocytosis. Other cells that are able to perform phagocytosis are dendritic cells. Dendritic cells perform phagocytosis as they wait for these microbes to invade the body, and then afterwards they send signal to the adaptive immunity system telling them that there is a microbial invasion. Other modes of attack of white blood cells are through secretion of toxin. Eosinophil, aside from being a phagocytosis, is actually able to also secrete toxin. For example, in the cases where the pathogen is too large to be ingested, the toxin that is secreted by eosinophil can actually kill this pathogen. Another leukocyte that can secrete toxin is NK cells or natural killer lymphocyte cells. NK cells, they can secrete toxins onto surfaces that have been infected by virus or also cancer cells. Another leukocyte that have a role in second line of defense is neutrophils. Neutrophils have enzymes that are able to kill the bacteria invading the body. Another form of enzyme have a role in the production of nitric oxide. Nitric oxide have a role in inducing inflammation, which is beneficial to fight off the bacteria or pathogens that are trying to invade the body. Another part or role of the neutrophils are the generation of NETs or the neutrophil extracellular traps. The NETs are able to disintegrate the bacteria trying to invade the body. We're going to continue with the non-specific chemical defenses against pathogens. This is where toll-like receptors or TLRs and NOD proteins are functioning to recognize PAMP or pathogen associated molecular patterns. They recognize the pathogens because they are receptors, and then they initiate a series of defensive responses. Host cells also release interferons or IFNs to non-specifically inhibit the spread of viral infection. Complement system or complement can be activated through several methods. The methods are classical pathway, alternative pathway, and lectin pathway. Ultimately, they indirectly triggers inflammation and fever to destroy microbes. As the complement progresses, the complement proteins can actually activate other complement proteins. One complement protein can activate other 50 complement protein, which in turn can activate other 30 complement proteins. This is a cascade of complement. A circular hole in pathogen's membrane is formed by MAC or membrane attack complex, which is an end product of a full cascade of complement. As previously discussed, complement indirectly triggers inflammation. Inflammation is a non-specific response to tissue damage. It exhibits several symptoms or signs, such as rubor, which is the redness in light-colored skin, Kaler, which is localized heat, edema, which is swelling, and dolor, which is pain. Inflammation in the body happens because the blood vessels in the body go through dilation or vasodilation. Vasodilation can be promoted by the presence of several chemicals such as bradykinin, which forms when there is a damage to blood vessels, and they are powerful vasodilator. And then prostaglandin and leukotrienes, which are released by macrophages. Prostaglandin are used to identify invaders that is invading the body. And then histamine, which are released by basophils, they are the ones that also cause vasodilation of the capillaries. Eventually, inflammation ends with tissue repair. However, in the case where undifferentiated connective tissue cells, such as fibroblasts, are involved, the tissue repair cannot be done as if when the undifferentiated stem cells are involved. So in that case, instead of the typical repair of the skin, for example, scar tissue will form. And then in the cases of cardiac muscle and some parts of the brain, since they do not replicate, tissue repair cannot be done. As a result, these tissues will remain damaged after a heart attack or stroke. Now we're going to move on with fever. Fever happens because of the presence of pathogens. Pathogens trigger the hypothalamus of the brain to reset the body's temperature to a higher temperature. This is essential to enhance the effects of interferons or IFNs, phagocytes, and also lymphocytes. The fever will persist for as long as the pathogens are present. However, as the infection continues, it will eventually become more under control, and fewer active phagocytes will be involved. Hence, the level of the pathogens will also be decreased, and it will reset the hypothalamus again, decreasing the temperature of the body back to normal temperature, which is around 36 to 37 Celsius. In the case where second line of defense is still not enough to fight off the pathogens trying to invade the body will initiate the third line of defense, or the adaptive immunity. There are several types of antigens, which are the endogenous, exogenous, and autoantigen. The endogenous antigen is the pathogens that are reproducing inside of the body. Meanwhile, the exogenous antigens are the pathogens or antigens that come from outside of the body. And now we're going to be discussing the processing of the endogenous and exogenous antigen before um, a series of other processes in the adaptive immunity continues. The processing of endogenous antigens start with the production of polypeptides by nucleus cells. The proteases then cut the polypeptides into smaller fragments of amino acids containing epitopes. Epitopes are the three-dimensional structure that make the antigen recognizable by the cells later on. The fragments are then moved to endoplasmic reticulum and bind with major histocompatibility complex, which are a cluster of genes. The fragments will bind with MHC1 molecules to form vesicles. These vesicles will then fuse with the cytoplasmic membrane of the cell, which will then display the complex of MHC1 with the epitope. Immune system is now able to detect the antigens that are being presented. The processing of exogenous antigen is different from the processing of endogenous antigen in the sense that antigen presenting cells, or APCs, are involved in the processing of exogenous antigens. Usually it's dendritic cells. Dendritic cells will perform phagocytosis to the pathogens invading the body. And then lysosome fascicles containing the MHC class II fuse with the phagosome, and then antigens are clipped into epitopes by the lysosome. The epitopes will then bind with the MHC class II, and fascicles then fuse with the cytoplasmic membrane. This will expose the MHC epitope complexes. After the antigens have been processed, we can say that the third line of defense have officially commenced. Third line of defense involves several cells such as T cells and B cells. The T cells each has a unique and specific TCR, or T cell receptor, and they make TCR only binds to their specific epitope. There are several types of T cells that are present in the third line of defense. The first one is the TC cell, or cytotoxic T lymphocyte. The TC cells are differentiated in a way that the cell surface have glycoproteins of CD8 and CD95L. The TC cells secrete perforins and granzymes that destroy infected cells. The second type of T cells are TH cells, or helper T cells. TH cells are differentiated in a way that glycoproteins of CD4 are present in the cytoplasmic membranes of the T cells. And then their function is to help regulate the activity of B cells and TC cells, and also secrete cytokines that regulate the immune system. Another type of T cells are TR cells, or regulatory T cells. TR cells repress adaptive immune response and prevent autoimmune disease, and are activated by contact with other immune cells. The TR cells also secrete cytokines. However, these cytokines are different from those that are produced by other T cells. The lymphatic system is heavily involved in the third line of defense, especially considering that B cells, which are present in the spleen, is involved in the third line of defense. B cells have BCRs or B cell receptors that form antigen binding sites. When bound with an antigen, B cell goes through cell division and secrete immunoglobulins. There are multiple types of immunoglobulins. Activated B cells will turn into plasma cells by means of interleukin 4. Um, there is a typo here. I did not mean to say B lymphocytes, but instead B cells secrete immunoglobulin, not B lymphocytes. The plasma cells no longer have BCR, they no longer have the B cell receptor, so they're different from the regular B lymphocytes. Not all B cells will be needed for the antigen that is currently present in the body. The B cells that do not have appropriate BCR will essentially survive the apoptosis. Apoptosis will only be gone through by the B cells that have appropriate BCR. The surviving B cells and T cells will then communicate with each other through the means of cytokines. Earlier, we have discussed that activated B cells can differentiate into plasma cells. Plasma cells are able to secrete immunoglobulins, and one kind of immunoglobulin is antibody. Antibodies have the same specificity as BCR. So, when bound to an epitope, antibodies induce complement activation, inflammation, neutralization, opsonization, agglutination, and antibody dependent cell mediated toxicity. Plasma cells can also change the type of antibody Fc region or stem to do class switching. Class switching is essentially when the type of immunoglobulin that is being secreted by the plasma cells is changed into a different type of immunoglobulin. The class switching is controlled by Th cells and it is irreversible. We are approaching the end of the third line of defense. The dendritic cells also communicate with the TC cell and then activates the TC cell to secrete interleukin 2, which triggers the cell division of TC cells. Activated TC cells perform clonal expansion or reproduction of memory T cells as well as TC progeny. The daughter of TC cells activate and produce more interleukin 2 and interleukin 2 receptors and become self-stimulating. This means they no longer need APC or the antigen presenting cells or even Th cells. They can attack infected cells on their own. Memory T cells are very important in cases of future infection by that specific antigen that have infected the body. The memory T cells persist in lymphoid tissues for months or even years in preparation for that specific antigen infecting the body in the future. This antigen needs to match the TCR possessed by the T cells. The memory response is rapid and enhanced when compared to the response of the first infection. Not only memory T cells are formed, but memory B cells are also formed. Memory B cells and plasma cells are formed through a series of processes conducted by Th cells and B cells. The plasma cells secrete antibodies or immunoglobulin, while memory B cells move to lymphoid tissues in preparation for next encounter with that specific antigen that was previously infecting the body. And that's it for my presentation for the immune response. This work of presentation was sourced from the book which is used in this microbiology class, which is the Microbiology with Diseases by Body System. It is written by Bowman in 2019. Thank you so much for watching this video. Have a good rest of your day. Bye bye.